Hi everyone, I'm Lucas Pereira, and I'm going to talk about machine learning for exploratory data analysis and model diagnosis in oncology. I'm a product engineer at PLOS AI, and the analysis that I'm going to talk about were made with the help of Mohamed Tarek, a senior project engineer at PLOS AI. First, to give an overview of the topics we're going to discuss, first to represent some context around the data that comes from the paper in the lower right, and also some definitions of the problems we're trying to solve and some goals we have with the analysis. I will also give a brief overview of theories and examples from the techniques we're using, namely clustering and dynamic time warping. And I will also, of course, show the clustering results on the data that we use, and then describe an NLME experiment that we conducted in the dataset, talk about some key takeaways that we have from these experiments, and in the end, show the script that we used for some of the steps that we're going to show. First, to give a summary of the data and problem that we are focusing on, the study cited here collated data from multiple studies in oncology with the main goal of benchmarking classical mathematical models in the field against real cases data. In total, the data includes almost 1,500 patients, all of them with at least 3 measurements of sum of largest diameters, a very common variable in oncology. And among these, 652 patients had at least 6 data points. And this subgroup is the main source of data for our experiments. From the paper, they implement and use multiple models from the literature in the field, but we focus mainly on the general von Bertalanffy model, which from this study and another one I will cite in the future, in general had the best performance. And on the right is shown the analytical solution for this linear ODE model using a reparameterization that I'm going to discuss more later. And the high-level goals that we have with these experiments is to first explore tumor measurement data via clustering with DTW. So using these two techniques are combined to gain more insights from the data we have. We're also going to be able to visualize large and diverse datasets. As I'm going to discuss later, there is a peculiarity with the data of SLDs that we are using. And also showcase the use of population stratification for model diagnosing when we apply the clustering and sample a new population from it. Now going to the clustering, it's asked beforehand to give a bit of context of categories of problems in AI. There's a big group called supervised learning when we're looking for input label mappings. So supervised learning are applications in which each data point has a input and label pair. And what they're trying to do is to train a model to receive the input information and try to predict the respective label. A very common example would be a neural network receiving an image and trying to process the contents of the image, trying to classify it in a set of categories. The other big group of applications in AI is unsupervised learning, where instead of data points having input and labels, you don't have labels for the points. And instead of looking for that mapping, as in supervised learning, you're rather searching for patterns and trying to gain more insight into the data. And inside supervised learning, a very big subfield is clustering, when we want to identify subgroups in the data. In healthcare, very common examples of these groups are responders and non-responders, as we will see in a few slides as well. But the idea of groups in clustering is much broader and more abstract, and you can identify groups that you wouldn't expect to be in the data, or maybe that the interpretation isn't so trivial. And the mindset that we have towards clustering in this presentation is as an alternative to heuristics. So in oncology, there are some standards to identify these SLD trends and try to classify patients into different categories. But some authors sometimes use heuristics to try to make this classification, and clustering would be an alternative to these approaches, which would be automatic and focus directly on the data without without introducing any type of bias through a heuristic that we created manually ourselves. And in clustering, a very popular technique is called k-medoids, which I included an example in the right. So going through the main steps in the algorithm, starting with in square 0, when you just have your data with no labels, you would then choose k random data points as cluster centers, with k being an integer that you choose in the beginning. And on square 1, 2 points in red were chosen as the cluster centers. And on the next step, you assign all other points to the cluster with the closest medoid. So in square 2, you can see that the remaining points were split into groups according to which red point is the closest one. And I assigned here different colors and also separated them in the figure. And in the next step, we then try to decrease total costs by swapping medoids with other points, if possible. So here what we are trying to do is to change which data points are used as medoid clusters such that the total cost is minimized. And the notion of cost in k-medoids is the distance of all points in a cluster to the medoid of that cluster. So as we change medoids, there are choices for medoids for each cluster, that these total cost or total distances should be minimized. And after making this change of which are the red points, the ones used as medoids, we will then update the assignments as in square 3 and make the separation again as in square 4. And these last few steps of changing medoids, updating the cost, and trying to minimize it would be repeated as necessary until the algorithm converges to stable medoids. And an alternative to k-medoids in clustering is called k-means, also very popular. But the main difference is that the centers of clusters aren't necessarily data points, but are rather defined as the mean position of all points in a cluster. And of course, in practice, that usually won't lie on top of a data point. But the main advantage of k-medoids is that it is more robust against outliers and noise, which are very common in real case data. Here I created an example to show how k-medoids can work in some cases. On the left is artificial data that I generated through 3 normal distributions, one concentrated at the top, one on the right, and another one on the bottom left. And on the middle, you can see the results from k-medoids, how we decided to assign points to the 3 clusters. And visually, it seems like a good splitting, judging by where the initial clusters were generated. And on the right is, again, the all points, but with the ground truth labels, as in knowing from the dataset originally which points were from which cluster. And one difference that's worth pointing out is that in the k-medoids results, there aren't really intersections between the clusters because it's, again, based on the distance to the closest medoid. But if you look at the right, there are some intersections, which you can notice as points of one color in the middle of a cluster of another color. And one last point that's also important to point out is that there are 2 clusters between the middle and right plots that got swapped, namely the blue and yellow ones. And this serves as a reminder that k-medoids doesn't pay much attention to which cluster is which, as in the labels that you get that you give each cluster. As you run the algorithm multiple times with the same data set, the labels of the clusters may be permutated. That doesn't change fundamentally the results of the algorithm. It still made the same separations. But if you're trying to interpret and give the clusters names and you want consistent results, it might be useful to fix which points are used in the first iteration to start the algorithm. Because they are chosen randomly, but if you always start in the same ones, the rest of the algorithm is deterministic and you can get consistent results. Before moving on to dynamic time warping, it's important to discuss a bit the broad idea of dissimilarity between mathematical objects, since this notion is really important for many numerical analysis, especially inside AI, when we want to train sometimes models that have the goal of minimizing some error or loss function, comparing the prediction of the model at some point in the dataset. So in the first line here, I just included an example between two numbers. It's very familiar to everyone. On the second line, now I want to define some type of distance between vectors. There are many ways to do this. Here, I just calculated the difference and applied the p-norm to the result. On the third line, you could say it's a bit of a fancier example. If you wanted to try to measure the dissimilarity between two probability distributions, P and Q, you could use the expression shown here, which is the Kullback-Leibler divergence. And on the fourth line is an example of a dissimilarity between two matrices, P and Q, in case that's necessary. And following this logic of trying to define the similarity between different types of objects, in the case of oncology, focusing on measurements of lesions and sum of largest diameters, it's really common for these trends for each subject to have uneven spacing in time and the sizes that you get, but also the amount of measurements across patients tends to vary a lot, depending on for how long they are on the treatment and how the treatment is going. So here on the last line, on the left, you would have a patient with 5 measurements and you want to calculate some type of dissimilarity against a patient that has only 3 measurements. And of course, both trends have uneven spacing and sometimes very different measurements. So in this last case is where dynamic time warping comes in our analysis. It is a method to compare two discrete sequences with the advantages that it is able to handle uneven spacing and also different sizes, which is really important in our case. And here in the image, I included a simple example between two sequences. The one on the top has 5 elements with uneven spacing, and the one on the bottom has 4 elements with uneven spacing. And what DTW is able to do is to calculate a mapping between elements on the first sequence and the second one, and also return a cost from, that results from this mapping, which of course we are going to interpret as a dissimilarity measure for our analysis. And now to show the results that we got combining chemioids and DTW for the data we're discussing. Here we can see that the workflow was able to identify visually distinct patterns using 3 clusters. The first one you could interpret as a fluctuating cluster where the trends don't change up, either change too much, either up or down. But on the middle cluster, trends generally go down, and on the third one, they tend to go up. And these labels and interpretations that I just presented aren't part of the workflow. As I said before, chemioids doesn't try to impose any meaning in the clusters or even the order of them and permutations. All interpretation and thinking about these labels and what each cluster means is still up to the user and has to consider, of course, the context of the data, the studies, what questions we want to answer, and the analysis we're going to run, the models we're going to use, and so on. And not only that, but the number 3 that we used as the amount of clusters could also be changed. We could have set to only look for 2 clusters. Maybe if we were trying to find patterns of only trends going up or only going down without much interest in a fluctuating trend. Or maybe we could also increase the number of clusters, say 5, 10, 20 clusters, how many we wanted. But again, always keeping in mind of the interpretability of all these clusters, what each one of them would mean in the context of the data, the experiments, the models we're going to use, and how useful all this information is for us to gain more insight about the data. And another aspect to tie to the paper is that the authors mention a standard in oncology to classify patients between responders and non-responders, depending on how the SLDs change in relation to baseline. And they comment that this standard mostly focuses on changes from baseline, and that's not really what they are after. Considering the models that they are going to fit, the linear ODE models for trends, and also the analysis they are going to run, they wanted a metric that would separate patients using a bigger picture view of the trend. So not only considering the change from baseline, but each measurement itself. And they, instead of using the standard, then they created a heuristic and tried to classify patients between 3 clusters. So as said in the beginning of the presentation, as an alternative to a standard that, depending on the analysis you're going to run, may be more or less useful from a numerical point of view, and instead also of creating heuristics from scratch manually, which could introduce some type of bias and influence all the remaining steps. You could instead use a workflow with clustering and DTW for this case and get this type of stratification in an automatic way, and that uses directly the data. Moving on to the NLME experiment that we conducted, we based the model on top of the classic Bertalanffy dynamics with the analytical solution shown here, which is a reparameterization from the citation below. We added on top of that random effects on the parameters, those being V infinity, which is the steady state total volume of lesions by the end of the trend of the patient, V0 representing the initial total volume, and omega showing up from the reparameterization done by these authors. And of course, lastly, we also included a residual error term in the model. And to fit this model, we actually sampled a new population from the clusters that we identified with the previous procedures by using 10 subjects from the up cluster and 40 subjects from the down cluster. And after feeding this NLME model to this new population, we then used it to simulate many different cases of this population. So we would have the information necessary to create VPC plots for the new population and each subgroup that we used here. And this is the VPC obtained for the entire new population that we used. Just as a brief summary, the upper blue band refers to the upper quantiles of the simulations. The middle gray band contains most of the medians of the simulations, and the lower blue band includes most of the lower quantiles of the simulations. And the black lines here refer to the quantiles of the data itself. And the goal of the VPC is to compare the model predictions and the data to see if there is still any big gap across the timeline, or maybe in some level of quantiles. The fit here is reasonable. Again, the goal here is not to create some state-of-the-art NLME tumor growth dynamics model, but rather to showcase the use of the stratification of population through the procedures we shown with clustering and DTW. Moving on to the down cluster, now we can see a bit more of a difference between the simulations and the observed data. But it's still a reasonable fit, not too different from the previous one, since the down cluster is predominant in this dataset. As a reminder, there are 40 subjects from this cluster and 10 from the up cluster. And now the main point of these VPCs, we can see that for the up cluster, unlike the two previous ones, it shows now some worse fitting for the population in this cluster, mainly indicated by the red region at the top, representing outliers. And this can be mostly explained by the fact that this cluster is underrepresented in the population, but also the fact that the bands expand towards the end of the timelines, since in the cluster, in the new population, there are patients with trends going up, others going down, and the model trying to fit both of these opposing patterns may create some uncertainty in the simulations in these points further down the trend. Some key takeaways that we can have from these experiments, first regarding the cluster stratification, is that the procedure was able to identify the existence of the subgroups without any prior standards, logics, or heuristics in place, just based directly on the data. And this is really important because it makes this procedure application agnostic. So if you were dealing with data of a different type in a different context or with different challenges, you could apply the same procedure with very small adaptations. And when you have the stratification of the population by the subgroups identified by the procedure, you can then use this to evaluate the performance of the model in each subgroup of the population. So not only checking the performance in the whole population, but seeing if there's maybe some population that was underrepresented or that the model had more problems fitting. And all of this mixes with the PLOS AI ecosystem of tools. As shown here, it can be used in an LME model for tumor growth dynamics, but also maybe as a pre-processing step when using PLOS, which is one of our tools with heavier AI usage. And now I will switch to the script that we used for some of these steps. In the beginning here, there's mostly some environment setup, bringing packages mostly for statistics, plotting, and so on. We download the data from the paper's website. And here I can give an overview of how it looked originally. The authors provided for some anonymized IDs for each subject in the first column, the days in which each measurement was taken, and the SLD for that measurement. But there are still some treatments needing for this data. For example, we can see that some time points here are negative, which is not really useful. So we will need to change that. And first, we renamed some columns here. We had to remove some problematic rows. Also take care of the types of some columns. We needed to shift here the days by the minimum to avoid those negative values I just mentioned. And after that, we restructured the data frame such that instead of this format here, where each line, every patient occupies multiple rows and each row is an individual measurement for that patient, we want instead each row for a patient and including a vector of the time points and the SLDs for that patient inside each row instead of everything spread out. Some more rows were needed to be removed here, and by the end we also created new columns. The measures column will be used to indicate the number of measurements for that patient. There's also a column for the diameters, the diameter trained relative to baseline, and also a column for the volume trend for the patient. And by running this block, we apply all these changes. And to see the new state of the dataset, now each row represents a patient with the time points as a vector, the SLD's trend here as a vector as well, number of measurements, diameters relative to baseline, and the total volumes as well. Moving on, we then started focusing on the chemidoids application, but we first need a symmetric matrix for that. The specific chemidoids implementation we're using actually requires a pairwise distance matrix to work with. So we use this function here, pairwise, which will iterate in all lines of the dataset and apply the function in this line here. So for every pair or every possible pair of subjects, it will calculate the DTW distance between the diameter trends. And we also set the number of clusters to 3. And here we run the chemidoids algorithm. It's fairly quick in this case. And we can also inspect the object returned. So here are the medoids chosen. These are the rows of the subjects that were used as medoids. There's also a vector of integers containing the assignments for each patient. So here there's only 1, 2, and 3 in this vector, indicating the cluster to which each of the patients belongs. There is also the costs for each patient, so its distance to the center of its cluster. There is a field called counts with the sizes of each cluster. So going back to the clusters that we got, there are this many patients in the fluctuate cluster. This is for down, and this is for the up cluster. So we can see that the fluctuate cluster is by far the largest. There's a total cost as well for this clustering, the number of iterations taken, only 5 in this case, until convergence, which is indicated here with the boolean. And with the assignments, we can then create a new column in the dataset containing that information. And then we filter the dataset to only include the patients with at least 6 measurements, since that's the focus of our analysis. And in the new state of the dataset, here you can see the new column at the end called assign, with the cluster for each patient. And this column can be used to then group the dataset into the 3 separate clusters. In this line, I just gave some names for each dataset to keep track of them, combine them all in the datasets variable, which is a dictionary mapping a key with the name to the data frame itself. So and here you can also see the size of each dataset, of course, with their names and number of columns as well. And finally, for the NLME experiment, we created a new population, just sampling first 10 subjects from the up cluster, 40 subjects from the down cluster, concatenating them and assigning to a field called mix in the datasets variable, which you can see here that it has shown up. And continuing this workflow, you could reproduce the remaining steps and analysis rerun as well, but these are the main points in the beginning of the workflow. Thank you all for your attention, and I hope to see you in future webinars. And in case there are any questions, you can all get in touch with us.